And now he's sort of accepting his style. He advised consultants. So he's at a university now, about 50 years old.
They're not, it was your wife who said I should come and sit by
Absolutely,
her.
absolutely.
Yeah.
I'll say a few words. Well, good evening, everyone, and welcome to this year's Haldane Lecture, and good evening also to those on the line and looking at this lecture on YouTube later on. It's great to have you all with us. And I'm delighted to welcome Professor Dame Kay Davies to join us tonight. Kay was an undergraduate at Somerville, then a graduate here, and then a junior research fellow here at Wolfson. In 1989, she went to what was then Green College as a fellow. She became Professor of Genetics in 1995 and a Fellow of Keble. And in 1998, she was elected as Dr. Lee's Professor of Anatomy at the University of Oxford Department of Physiology, Anatomy and Genetics. And she's now Dr. Lee's Professor of Anatomy Emeritus and co-director of the MD UK Oxford Neuromuscular Centre. Her research interests lie in the molecular analysis of human genetic disease, particularly the genetic basis of neuromuscular and neurological disorders. She first became interested in muscular dystrophy nearly 40 years ago, and many of her research group are dedicated to finding an effective treatment for Duchenne muscular dystrophy. She also has considerable experience of biotechnology companies as a conduit for translating the results of experimental science into new therapeutics and diagnostics. She's co-founder of Summit Therapeutics and OxStem.
Yep.
In 1999, she set up the MRC Functional Genomics Unit, aimed at exploiting genome information for the analysis of the function of genes in the nervous system. In 2000, she co-founded the Oxford Centre of Gene Function with Professors Ashcroft in physiology and Donnelly in statistics to bring together genetics, physiology and bioinformatics in a new multidisciplinary building which was completed in 2003. She has an active interest in the ethical implications of her research and in the public understanding of science. She's a founding editor of Human Molecular Genetics and a founding fellow of the UK Academy of Medical Sciences. She's a fellow of the Royal Society and a member of the European Molecular Biology Organisation. She became a governor of the Wellcome Trust in 2008 and was deputy chairman from 2013 to 2017. And finally, she was made Dame Commander of the British Empire for services to science in 2008. So please join me in welcoming Dame Kay Davies.
Well, it's a privilege to be standing in Wolfson College. When I was a graduate student here, I just recovered from doing an undergraduate course in chemistry, and I decided that biology was much more interesting. This will make Angie Russell smile. And from then, I was introduced to geneticists, and having the experience of a graduate college had a large impact on me, and having the JRF to follow so that I could really build up the research momentum in genetics and think about clinical medicine, as I said, had a great influence. So thank you, Wolfson. I've been very busy since then. I don't come in that often, but hopefully I'll be able to come in more. I should also say one word about Haldane, and the Haldane family, as many of you will know, were very, very talented. JBS Haldane was the first to demonstrate genetic linkage, so I'm really standing on the shoulders of giants here. He really did the unification of Mendelian genetics with Darwinian evolution and natural selection. He helped introduce population genetics and methods of measuring mutation rates. And all of those are relevant to what I'm going to tell you about today in my journey. So, genomics and rare diseases from gene to clinic. During this lecture, I hope to persuade you that just the study of some of these rare diseases is going to make a huge difference to all our lives, more than just the impact on those rare genetic diseases themselves. But to begin with, since I've got the word genomics in the title, I thought I'd better define what a human genome is, because I don't want to lose people in slide number 2. You all have a genome, and it's exactly the same in every cell in your body, and it's how the cell uses that genomic information that makes the difference between a muscle cell and a liver cell. The holy grail was that if you could work out the whole human genome sequence, that is how you put those letters together, which are bases A, C, G, T, in a code that tells what proteins can be made, then we'd be able to understand a lot about evolution and a lot about the diseases. Oops, sorry. So the first draft human genome sequence was produced in the year 2000, I'm embarrassed to say, because 26 years later. So this was heralded as a big breakthrough. And I just thought it would be interesting to go through 2 particular quotes with you, one of which comes from Francis Collins, standing on the right there, that headed the National Institute of Health. And he said, decoding the human genome was the most significant undertaking that we've mounted so far in an organised way in all of science. That bit is true. But whether you're judging history as more significant, even splitting the atom or going to the moon, I'll let you judge that when I get to the end of my lecture. Maybe we are moving in that direction. And then Mike Dexter, who was then CEO of the Wellcome Trust, said this is an outstanding achievement, not only of our lifetime, but in terms of human history. So there is no question it is transforming clinical medicine. I think the only problem is the timescale. We said we'd have cures for a lot of these diseases within 10 years. I even said I'm going to cure muscular dystrophy before I retire. Well, I haven't retired yet, but I'm getting close. And I think we're some way down that journey, but we haven't reached the end. So, in 2000, we had the genome sequence. What on earth does that mean as far as the genes? What comes next? And I think this is where rare diseases really come in. And they affect an awful lot of people in the world. 80% of rare diseases have a genomic component. Half of those new cases are found in children. 7% of people are affected by rare diseases in their lifetime, so there may be even people in this room. And within the UK, 3.5 million people are affected if you consider the patients and their families. That's a huge impact on their lives and a huge impact economically, although I don't want to measure it in economics. So my story will centre around a particular rare disease, which is more common than most rare diseases, called Duchenne muscular dystrophy. Here the boys go into a wheelchair before the age of 12. They used to die in their teens, and they now survive into their 20s or 30s. That's good clinical management. It's got nothing to do with genetics, but it does have a lot to do with diagnosis. If you can spot what's wrong, you can think about how managing the patient. So I'm going to talk about the importance of diagnosis, how DMD and the study of the research that's gone on in DMD has really developed genetic approaches for treatment, which will affect the treatment of other diseases, and then the need for newborn screening. So this just says what I've just said. This only affects males. It's an X-linked progressive muscle wasting disorder. I just click it all the way down to the bottom. But there still isn't an effective treatment, and certainly we're a long way from a cure. But the interesting thing about this disease, which is why one of the reasons I decided to study, it's got one of the highest new mutation rates in any genetic disease. So unless you can screen every pregnancy, you'll never eradicate it. And that has something to do with the fact that the gene is very large and factors we don't yet understand. And so of all the diseases that need a therapy, this is one of them, because we're never going to, I think, for this particular disease, be able to screen every pregnancy. And of course, screening pregnancy is not acceptable to everybody in the world either. We have to think of effective treatments. So what does it do? That looks a pretty complicated figure of muscle bundles at the top. The only thing you need to remember, this is looking at the front end of your muscle fibers, and you can see the white staining around the edge of that normal muscle biopsy is the protein that's very important in this disease called dystrophin. It was identified by an American group under Lou Kunkel, but the most important thing is that all patients, the DMD patients, forget about the middle slot, have no dystrophin in general. And because of that, their muscle doesn't function. And this only becomes important at birth, and I'll come back to that in the end. The middle frame is a milder form of the disease called Becker muscular dystrophy, where the body produces a slightly smaller protein often that doesn't bind as well. That's why it's not quite as white at the membrane. So dystrophin, this looks a very complicated slide, you just have to watch the red line. If you take that away, that link between the outside cell and the inside of the muscle cell is destroyed. That allows enzymes in to destroy the muscle. And if you like, it's a bit like a shock absorber. You stretch your muscles and they should align. If you don't have dystrophin, they gradually misalign, and eventually you get the muscle degeneration. So, as I said at the beginning, this is perhaps one of the hardest diseases to treat, because we need to get at least 20% of the wild-type levels to have any clinical effect. We need to target both the heart as well as your skeletal muscle. Muscles make up 40% of your body weight, so to be able to target all of that muscle is a real challenge. And you need to avoid any immune response, because most patients haven't seen dystrophin before. If you put dystrophin back, the body may produce antibodies against the dystrophin or against anything that you may be introducing to the body to deliver that dystrophin. And you need to do a lifelong treatment. So the development of treatment depends on your understanding of what's known as the central dogma. You've got the genome, and with DNA, the ACGT that's transcribed into RNA, which is then translated into the protein that differentiates between your liver cell, muscle cell, or whatever. And so in order to devise several treatments, you can tackle any part of this journey. And the first is gene therapy, and there's been a lot of gene therapy in the news. This is where you take all the nasty things out of a virus and put your gene of choice in the middle, but leave enough machinery in the virus so it can invade a cell and reproduce and produce your protein. The problem for Duchenne muscular dystrophy was the gene is one of the largest known in the human genome. So it wouldn't go in to the mostly used virus known as AAV, assisted adenovirus vector. But this is where the great joy of human genetics is that it's international and it's collaborative. And so you start searching all over the world for patients that might give you a clue about how you might solve this problem. And this gentleman actually came from Birmingham. The late Sarah Bundy sent us this sample. You can see him at the top left there, where he's in the army. He's just walked up that cliff in the bottom left, but you can see he's beginning to walk a little bit like this in the bottom right. And so we thought he must have a little bit of his gene missing, so the gene wasn't, we could produce a smaller protein. But we were astonished to find that he had 50% of his gene missing. It just goes to show nature is full of surprises. And that meant that if we could demonstrate that he didn't have some compensatory effect, we had the possibility of producing a smaller protein, because we could design something that would go into that virus, which was much smaller. And this looks a little complicated, but if you just concentrate, this is that white that was a dystrophin on the edge of the membrane here in a normal individual. This is the central part of the protein. This is this patient's DNA, muscle biopsy, and you can see the abnormal muscle fiber distribution. There's nothing in the middle, and the carboxyl terminal on this end, he's back and he's got the muscle fibers testing positive. So he does produce a smaller protein. So that meant that the truncated gene could open this way to treat muscular dystrophy. And this gene is now the foundation of all gene therapy trials in the world, because we can actually use molecular biology. This is the full-length schematic of the protein, and this is the mildly affected patient. I went to his 65th birthday party, and he died in his early 70s. He has a distant cousin who's been very cooperative and given us samples. He also is now in his 60s, and he is weak in his muscles, but it's certainly not affected his life very much. Now, using a lot of genomic engineering, which I'm not going into, there are now lots of companies that have designed different mini-genes based on this individual patient that we found all those years ago. And not only that, they've actually initially injected them just into muscle, but they've now introduced them by intravenous injection of the virus. So now the gene therapy gets into the heart, and it gets to some, but not all, of skeletal muscle. Now, we don't understand the reason for that, and the pioneering company that has done this is Sarepta. So their gene therapy was approved 3 years ago, and it significantly shows disease progression, but it doesn't cure it. And again, I think we need to understand a lot of the biology. I was there at the FDA conference that approved this therapy, and there were videos of children that had had the therapy that got up and ran up the stairs, and there were children that had the therapy, there was no effect at all, and we still don't understand the difference between those two. Now, it doesn't mean it's not a useful therapy. It certainly is, because on average, you will get a response. But what we need, 3 deaths reported, is because the virus is toxic and we have to put massive doses in. So we need some very bright genetic engineers to come in and tell us how to make better viruses. But that will happen in the next 5 years. And the other driver of this really is, when I started this, no single company was interested at all in Duchenne muscular dystrophy. Suddenly, once treatments looked as if they were going to be in the horizon, then the Novartises of this world, the Roches of this world, the big pharma have moved in. And that's really helpful, because that means they've got the ability to set up the right, these were the wrong trials, but bigger trials and better understanding of the biology. Still, we have an immune response to the virus, so we're still a long way from delivering what we really want to do to all muscle, and to get rid of the cardiac involvement as well. This therapy will treat all patients. It doesn't matter what their mutation, which bit of the gene they've got missing, they all don't produce dystrophin. So if you reproduce dystrophin, you will theoretically cure the disease. And I'll come back to that at the end, because the therapy that I'm currently developing with Angie Russell is, again, trying to compensate for the lack of this dystrophin protein. I've already said that. Oh, I should say, each one of these therapies, gene therapies, costs more than $1 million per patient. You only have to do it once. But again, most health services in the world have not got used to the idea that they are going to be saving lots of money and that they need a new model of how to actually treat some of these patients. So they pay up front and they have to pay it back if the therapy doesn't work. And I think we're sort of in that transition now with regulatory authorities to get some of these most effective treatments to the clinic. But there is good news. It's not Duchenne muscular dystrophy, but in eye diseases, I mean, this slide came from Robin Ali from London 7 years ago, and they were already curing retinitis pigmentosa by putting replacement genes back into the eye. And the eye is a very privileged place for the immune system. You don't get rejection, and you can inject it to the back of the eye. So how will it affect you? Well, in 2024, they're now developing the delivery of a growth factor that slows down macular degeneration. So I hope they get that soon, because I'm not suffering from that yet. But this is something that the GP might be able to inject into the back of your eye. If you can make it cheap enough and it appears to be safe, then I think we're entering a new era. And that is a technology that was pioneered through this rare diseases research. The other part of gene therapy is called gene editing. And here, instead of replacing the gene, you correct the mistake. And I'm not going into the ins and outs of how you do that. The Nobel Prize was won by Charpentier and Doudna because of the development of this technology. Since those two ladies, I'm pleased to say, who had Nobel Prizes, was developed, there have been lots more generations of CRISPR. And so it's faster, it's safer. And so you can go in there, correct the mistake, and it's a one-off treatment. So you can imagine that's a lifelong treatment. So just recently, a young baby, obviously a young baby, a 6-month-old was diagnosed with an enzyme deficiency in the liver. He was too fragile to be able to do a liver transplant. And so the gene technologists in the University of Penn, together with the clinicians, got together with the FDA and designed a personal CRISPR delivery to try and cure this baby. And they give the KJ, as it's called, 3 administrations. And this time they didn't use a virus, they used another vehicle, but the same principle. And it was just amazing because this baby is now doing very well. It doesn't have any of the mental degeneration that you could have predicted it would suffer from. So I think we're looking at a revolution here where you can actually start thinking about correcting your genetic defect. And not only that, the FDA in the last couple of months have decided they approved this in 1 week. So that's quite extraordinary because they could see the promise. And now they're coming out with something called the plausible mechanism pathway. So if you can prove that it's highly likely that your treatment will work, they're willing, and you've got good biomarkers so you can test how well it's working, they're willing to consider giving permission for a trial in that disease. That in itself will revolutionize people's attitude to how you might treat disease. And that means any disease, including cancer, diabetes, not just rare diseases. So that's the genomics. You can replace the gene, you can alter the gene. I'm now moving on to transcriptomics, as it's called, where you're actually transcribing that DNA and genomic information into RNA. Can we interfere with that in order to think about treatment? Most genes are made up of lots of different pieces, and the DMD gene is extraordinary because it's made up of 79 different pieces. And what happens in patients is that if they have a deletion of one of these segments, which are, as you can see from the top, fit together very neatly when the machinery in the cell gets going, if you take one of those segments out and you don't get a matching thing here, then you'll get non-translatable this RNA can then not make protein. So the principle then of treating the disease at the RNA level is known as exon skipping. If you take out another segment, then you can marry up the ends. And you can get a new message in RNA, and then you get translation into protein. It'll be slightly smaller because you've missed more out, but it'll still be quite functional. So the principle is that you can do this for any sort of mutation across any sort of deletion in the DMD gene. And you can change the reading frame, so-called, and make the RNA translate into a functional protein. And because we understand a lot about what governs that exon skipping, we can think about how to hide the signals in the cell that promote that type of skipping. So if you hide the sites that induce the splice, then you can skip the exon. And this slide, don't bother to read it, just shows you that the FDA have now approved several of Sarepta's antisense oligonucleotides for the treatment of DMD. But you need to know exactly what the mutation is. So you have to sequence the genome first, understand what's missing, and then work out how to stitch it back together again. Now, I put non-approved in Europe because this particular approach works in slowing down the disease, but again, it's a long way from a cure. We don't understand how to target all the muscle efficiently enough to give you a real clinical impression. So non-approved in Europe, it's not really worth the price at the moment. That balance is not there. So we need more efficient skipping. Having said that, exon skipping doesn't work so well in Duchenne muscular dystrophy. In diseases like spinal muscular atrophy, which I'll introduce you to briefly towards the end, this is floppy baby syndrome. When I first started to work on SMA, these babies used to go home to die. If you get them early enough, you can skip their mutation and they can be almost well. So in some cases, it is proving to be almost curative. You can also use these antisense RNA oligonucleotides to silence genes. There are a lot of neurodegenerative diseases which have toxic proteins. If you can stop the toxic protein from being produced by stopping it at the RNA level, then essentially you can cure the disease. So finally, to the end of this, protein. Some 15%, as the next slide will say, of DMD patients have a point mutation, which means they don't make the full-length protein. It stops, and you get incomplete translation, and the RNA degrades. By screening with a variety of chemicals, you can persuade the cellular machinery, to continue to read through that stop signal, and so you get a functional protein. Again, this has been pioneered by PTC Therapeutics. They've introduced this drug. It has been approved by the FDA. It has been approved in Europe, but it isn't very efficient again. And so I think that tells us again, the real challenge is how do we deliver to all that muscle for a disease like Duchenne muscular dystrophy? But just to broaden it a little bit, 10% of inherited diseases are caused by stop codons. So if we can find other chemicals that would read through their stop codons, we may go some way towards finding an effective treatment for those diseases. So, can we do anything about that protein? As I said to you, DMD is caused by the absence of dystrophin. It's a bit like a shock absorber. We were very fortunate to be trying to identify both the dystrophin protein, but also something that might be similar to it. And something that we call eutrophin is another protein in the genome. It's big. And you don't have to be a molecular biologist. This is dystrophin with all its units, and this is eutrophin with all its units. Eutrophin slightly smaller, but you can see it's very similar indeed. And in fact, for those binding sites that are really important for function, it's 90% similar. So there was an opportunity here. Are there ways in which, why doesn't nature just make more utrophin? So we looked at the analysis in patients. What happens in patients? And the clue came from early on in development. This is dystrophin. There's the white at the edge of the membrane. And as you go through 12-week, 16-week, 20-week, 32, and the 9-month infant, you can see dystrophin at the membrane normally. Utrophin actually co-localizes with dystrophin very early in human development. In fact, I don't know which one comes first, utrophin or dystrophin, but it's clear that it's almost acting like a fetal form of the missing dystrophin, and then when you become an adult, you only find utrophin at the neuromuscular junction. And we know now, through our studies, that this is because it's controlled. The cellular signals that tell you whether to produce dystrophin or utrophin are there in the adult, switched on in the adult, and therefore you only get dystrophin at the membrane. However, if you damage your muscle, utrophin reactivates, builds the muscle again, then is downregulated, and dystrophin comes in to take its place. And what happens in muscular dystrophy patients, of course, there is no dystrophin to take its place, so the muscle degenerates. So utrophin in that same cartoon. And investigators other than ourselves have demonstrated you can cure a mouse by increasing utrophin, and you can cure a dog model by introducing utrophin, either a full-length utrophin or one of those mini genes, very similar to the dystrophin mini genes that I mentioned. But you need to be able to do that in a much more intelligent way. And so the reason Angie Russell is sitting in the front row is because she's a fantastic collaborator that I've been working with for almost 30 years or something on this program. Well, I'm not saying that to embarrass you. I think it's just, it takes that long to get to almost the end point. How do we set up a transcriptional screen? And this plays to the importance of multidisciplinary research. I couldn't do this if I did, even though I was originally a chemist. I certainly don't know medicinal chemistry in the same way as the chemists do. The other revolution is that you used to have to screen millions of compounds. That was incredibly expensive. I'm sorry about the simplistic Angie here, but if you now, you can start getting an enriched library to screen against your cell line and ask the question, does this chemical increase utrophin or not? And the beauty of this approach is that it is not going to be mutation dependent. It'll cure all patients. And with a bit of luck, it'll be cheaper than the 2.6 million mark that you get for gene therapy. So where are we? Well, the first step is you can't do a clinical trial without setting up a company to take you to that clinical trial stage. So if there's anyone in this audience who's got a really good idea to do something clinically, you really very early on need to get your collaboration with your clinicians, and you need to think about what will industry want in order to be able to take this into the clinic. And we were very fortunate to be able to, with my husband then, to found Summit. And it was their expertise that allowed us to take the first chemical compound, which we called azutramid, into the clinic. And I'm just going to show you the result of a phase 1/2 trial. You don't have to look at the complexity of the trial design. All I need you to concentrate on is here. This blue marker here is a marker called embryonic myosin that indicates that the muscle is regenerating. So if you've got healthy muscle, you would hope that it doesn't have much blue. So you can see there's the baseline, lots of blue, not very healthy muscle. And after 24 weeks of treatment with azutramid, these patients were demonstrating that they'd increased utrophin and they would decrease degeneration. The problem was, at 52 weeks, which is our final readout, we had no effect at all. Now, that was obviously incredible. This shows you have to have grit. You have to believe that that really worked, and there may be a sensible explanation for this phenomenon. And don't have time today to go through all of that, but suffice it to say, we know that between 24 and 52 weeks, there's a lot of metabolism of this drug, and therefore it isn't in the muscle anymore, and hence its effects gradually fade away. Now, industry did not believe us when we said that. That's just our wishful thinking. So Angie's group then went on to find out where was isotretinoin binding, how was it acting, and it turns out that it binds a receptor called AHR, which is very important, and if you get other chemicals that are influenced by AHR, it's an antagonist, then you also increase utrophin. So our job now is to find other antagonists like isotretinoin, but without the bad effects of isotretinoin, without the turnover of the drug. And without being too optimistic, I think we're down to our last 2 molecules where we hope that we will have achieved that goal. And again, then we have to set up a clinical trial to take it into the clinic. We can also, since we have one mechanism to increase utrophin, there may be other mechanisms that are acting just like they do in early fetal life, and we may have the opportunity to develop other chemicals which we can use in combination. It'll be used to treat all patients worldwide simply by popping a pill. But of course, it will have side effects, as every pill does, but not necessarily big ones, as GLP-1s demonstrate, obesity drugs demonstrate. There are certain drugs where you can certainly manage the side effects. Is this a type of approach applicable to other diseases? And the answer is yes. There's a disease called sickle cell anemia, a blood disorder, and Vertex have just developed a CRISPR-based therapy where they alter one nucleotide to make the fetal form of hemoglobin persist in adult life, which compensates for the mutated adult globin. This will revolutionize the treatment of sickle cell anemia if it didn't cost so much. And sickle cell anemia is particularly prominent in Africa. So again, we have to find a mechanism of trying to get some of these treatments more broadly applied. So just to summarize, genetic approaches to DMD in 2026, I think there's a lot of promise, but there's a lot of new developments that we have to make to get around the immune response and to make it more efficient. I'm hopeful that the modulation of utrophin will help, and I don't mind if I get there first, second, or third, because I think it'll all be a combination. But as we get to this point, people didn't want to know in their families that they had DMD because they felt it disturbed their interaction with their children up to 5 years of age. Once you have an effective treatment, that balance shifts, because the earlier you can get the treatment in, the more effective it's going to be. And I think that's where we are. And coming back to spinal muscular atrophy, which I worked on in the early 1990s, there are 3 types of the disease, depending on mildness, depending on how much of the protein that you produce. Type 1 is a devastating disorder. Babies are born very floppy. And as I said, they were sent home to die at one point. In fact, Victor Dubovitz and I went up and down this country, the UK, talking to neurologists about getting samples to try and find out more about this gene and where it mapped so we could isolate the gene. Just by increasing awareness of this and the possibility of a diagnosis, we doubled the frequency of SMA in the UK population. So what can we do? Well, there is now newborn screening for SMA in Belgium, pioneered by Laurence Servet, who's now in Oxford, fortunately for us, at the Neuromuscular Centre. So this is Jack. He had no symptoms when he was born, which is very often, well, it isn't very often, it's sometimes the case. And because he was treated very early on, he's now walking. Some of you may have read the news about Jessie Nelson, who had twins suffering from SMA. They were diagnosed a little bit later and therefore will probably be in a wheelchair, but at least with the new therapies, which is gene therapy, replacing the gene, or exon skipping over the mutation, we can actually essentially arrest this disease and give these children a normal life. But we have to do newborn screening. So there's a lot of pressure on the government to do this. I know you don't need to know that. Cancer is also a disease of the genome. I think I just mentioned this in passing. We now know that every tumour is different, but we're able to identify certain pathways that are predominant in certain cancers. I'm just going to show you one, actually quite old slide now. One drug, vemurafenib, which is mutated in 60% of melanoma patients. If you treat, you can see the dramatic effect on the tumours. Now, eventually, the tumours find a way around this, and you need a subsequent different drug to keep the disease in check. But by understanding cancer pathways at a personalised medicine level, you can begin to hold the disease in check. This looks a very confusing slide. It's merely meant to show, because of genomics, we can look at signals that will tell us about a particular cancer and the tumour and then what it expresses. Its signature, if you like. And I've just put here, this is taken from the Australian paper in 26, 27, 28. These lists of genes are growing, so we can identify different cancers very rapidly. But perhaps even more encouraging, the first positive randomized trial of genomic-guided cancer tailored has recently been published. And you can see what a dramatic difference it makes to survival. So if you have the tailored treatment in blue versus the standard treatment in brown. And this is the way of the future. Now, coming back to the message, we're in a very exciting era. I hope you feel that. This slide, since I was going through my back slides, when did I last give a lecture like this? I was amazed to find that the role of genome information across the continuum of health and disease was published in 2011. And we haven't progressed because we haven't worked out an efficient way of getting it into our health systems. And we have to find a way of doing that. But genomics and rare disease, I know we're improving the lives of patients. We've got rapid diagnosis. This government has actually introduced with Genome England a pilot study of sequencing every newborn. We're picking up mutations. We're identifying new diseases. We can't always have a treatment, but just having a diagnosis, just imagine you don't have to take your child for these test after test after test in the clinic. You can say, this is the mutation, this is what's causing the disease, and that's the beginning of the development of an effective treatment. Better understanding of disease mechanisms means we can have better management of clinical journeys. And every time you open, you don't, but I do, Nature and Science, there's a new technological advance. So that means better quality of life for patients and their families. And I think that will happen in the next 5 years. And we'll have to just shout for it for everyone, not just those that can afford it, everyone. And I think that has to be the goal. So I'll leave you with the message. I think we're in a very exciting place. I know we're not being too optimistic. I think in the next 5 years, I'm going to have to spend a lot of time trying to steer government policy and facilitate getting some of this to the clinic. Otherwise, we really will never get there. And it's there for the asking. Even the diagnosis is going to make a big difference to patients' lives and their families. So with that, I'm happy to take questions. Thank you.
So we've got a lady with a microphone up behind us. So would anyone like to start with a question? Let me start then, and others can do that, which is this question of getting these treatments into the NHS, and you say we need to find a way. Is that fundamentally about finding a financial way to get investment up front because it saves money in the long run, or what is the practical problem getting it into the NHS?
I think it's training, actually. It's making, it's not just GPs, it's everyone within the NHS. I think we need to retrain some people in the use of AI so they can do these sequencing technologies. And that's one thing. We do have to talk to the regulatory authorities about you pay up front for a particular treatment, but you're going to get 20 years of effective treatment. So that's a huge cost saving for the NHS. So it's a new model. And these things are being discussed. I think we just need to push it a little harder.
Questions from the audience? Yes. We'll just, Edith, Edith, we'll just get a microphone for you.
We can usually hear you, Edith, but with online people can't.
Yeah, okay, I was just wondering, how far away do you think we are from whole genome sequencing of all new life, of all new babies?
I wish I would say it was very close. I think the pilot experiment, it's not experiment, the pilot study that the government are doing with Genome England will demonstrate, I hope, that there's a big yield in diagnosis for that, which will help families a lot and help their clinical management. Once we've done that and they can show it's cost effective, every baby will be sequenced at birth. And not only that, the sequencing companies, there's another technological advance last week where they can do it much more efficiently. That's going to make it cheaper. We've gone from $3 billion down to $100 a genome now. So I think we're in the right area. We can afford to do it. We've just got to make sure that somebody can interpret the data to translate it to the patient. So I remember we developed the first prenatal a very royal we, because we were collaborating, developed the first prenatal diagnosis of DMD. And we could do it in 24 hours. The problem was even the NHS practitioners couldn't actually coordinate. It still took them 3 weeks to get the results back to the patient. You can't, we've obviously advanced a lot since then, but those are the sort of challenges we have to think about. And everyone in the NHS is overworked, incidentally, which is why I think somehow we have to increase morale, educate them more, and give them the ability to participate in the revolution, and don't leave them behind.
Up here.
Thanks, lovely talk. I work on rare disease as well, and one of the challenges that we face is recruiting enough patients for clinical trials. Do you have any suggestions, or what has worked for DMD to have patients ready for trials? Because there is a demand, but clinicians might not just even have patients ready for trial-ready patients, essentially.
That's a very good point, and I skipped over it. It was on my first, second, or whatever slide. You need registries of some of these rare diseases so that you can, every time someone gets a diagnosis, they're put on a register. So it doesn't matter where they are in the world, because if it is rare, it may be you only have 10 patients in the world, but you can still do your clinical trial in different centres. But you have to have registries. And of course, for DMD, I was very fortunate in having lots of there was Muscular Dystrophy UK, there was the Muscular Dystrophy Association in the States, and we all worked together. This endeavour is international, and I think that's the way you recruit your patients and identify them.
The NHS has been mentioned quite often. I think one's got to be a bit fair to it, in that in 1948, 3.5% of gross domestic product gave you state-of-the-art for the people.
Yep.
Now it's 12 to 15%. No government has had the guts to discuss this with the people as to how it's playing. And there is a tendency, inevitable, for medicine to be pricing itself out of the market, because your surviving muscular dystrophy patients will go on to get coronary artery disease, aortic stenosis, cancer, and ever more expensive treatments.
No, that's true. But, well, I think if you treated, I'll take an extreme view here, if you treated a DMD patient so their lives are almost normal, they'd be the same risk as any other individual within the population. DMD is exceptional in the sense that you could never eradicate it. So I do believe that we still need always to chase a treatment for DMD. Whether we ever get to a stage where we're screening every pregnancy, so you eradicate, that's a bit, and it's not acceptable for an awful lot of populations around the world. On your NHS point, I think it's incredibly important we don't demoralise the NHS. And I hope that we do think about working in partnership with people to enable them to share in our ability to do some of these things. If we don't do that, we get a lot of dedicated people that are working in the NHS disaffected about what they're doing. We mustn't develop a parallel universe. We have to try and involve everyone, which is incredibly ambitious, but personally I think is worth the goal.
Any good treatment has side effects, a pill, X-ray, and so on and so forth. What are the side effects or the risks that genetic screening or genetic editing have?
Okay, no treatment was without risk, and the reason gene editing has not taken off as much as it might because of those risks. I mean, Vertex were able to do it for sickle cell anemia because they took the blood cells out of the patient, gene edited them, make sure there wasn't any abnormal things in the cells, and then put them back into the patient. I think we are still a long way of just injecting something that is going to CRISPR edit your genome in every cell, because I think you are likely to get off-target effects that you don't expect. So you're quite right. A lot of these things are going to come with quite serious side effects that we don't yet understand. But it doesn't mean they're not insurmountable, but that will take time.
We've got a gentleman here.
Thank you. You mentioned RGX-314 in connection of treatment for macular degeneration. Could you just explain a little bit more how that worked? I didn't quite follow the point you were making.
Well, I'm not an eye expert, but it's a growth factor. So that's all I can tell you. I'm just, and I didn't use ChatGPT to get that either, but it affects the way the cells. I'm not an eye. I don't know anything about eye disease, so I'm not even bothering to. It's a growth factor that will increase the health of those cells. It's all I can tell you. And a direct injection of that originally was a treatment for macular degeneration. If you could do a permanent fix so that you had constant production of that protein, it would make it more efficient. That was the point I was really making.
Is it in use now?
No, but it's. I mean, that paper was that clinical trial was 25, so it won't be long before it is.
Right. Okay.
Jagdeep at the back.
Can I just come back to the question regarding cost effectiveness? And so it's not the regulators, it's NICE that determines that. And NICE assumes that per quality adjusted life year improvement, that society, we the taxpayer, will pay £20,000 or £30,000. So for gene therapies that can cost 3 to 4 million per patient, it's very hard to get to that number.
Yep. Yes, I agree. And so what you need is, one of the problems is manufacturing viruses is expensive. I mean, you have to pay for the development of these drugs, but putting that aside, if you could get more efficient virus, so instead of using 10 to the 13 particles, you use 10 to the 5, it would make it a lot cheaper per patient. And that requires a lot of engineering to get there. We're some way away from that. Maybe we can get rid of viruses altogether and find other ways of delivering genes to the body. And certainly companies are experimenting with so-called nanoparticles without the virus, without the side effects. I don't know whether they'll be cheaper, But I think we have to think about cost. There's no point in just developing more and more esoteric treatments that are not going to... Is it really worth the human race doing that if very few people are going to benefit and not benefit very much? And I think we have to keep that in mind all the time. And at the beginning, when you start your research, you think, what patient population am I going to treat? How will it be? What is its likely price going to be? Because that's going to be part of the equation. It's very important.
Oh, one last question here.
So you mentioned that there's the higher de novo rate of mutations for this condition. And is that just like proportional to the length of the gene being so long? Or is that more than you'd expect even with that? And any ideas why?
It depends who you talk to. Most people say it's a very big gene, it's a big target. But that doesn't explain why 60% of the mutations occur in only one place. And it doesn't explain why they're mostly deletions. So there is something in the biology of the genomic biology that makes that more susceptible to deletions, and we don't yet know. We've sequenced it every which way, and we still don't understand why. Something's driving that mutation rate.
Okay, thank you very much indeed. I mean, I think for all of us, this was both a fascinating insight into DMD itself, but also the scientific journey and the way in which one collaborates in order to solve big problems for the whole of society. So on behalf of all of us at Wolfson and the audience tonight, thank you very much indeed.
Careful I don't go off with your apparatus. I think maybe we can see where it's attached. There we go. Thank you very much.